on. And so this is nucleic acid methods. All right, so here we're seeing the nucleic acid methods, and it has a nice demonstration of clockwork because most of them actually work a lot similar to an industrial process or a clockwork process, right? It's very mechanistic, isn't it? Okay. And so again, the learning goals, outlines, all that good stuff. This is the exploration of your gene is going to be rely on key tools. We have the rapid progress in our biotechnology. This is actually primarily the results of a few key techniques, and these are the few key techniques that have been developed. There's actually a really interesting story about PCR that I'll tell when we get to PCR. So don't go to sleep yet. Now, we have made incredible progress in biotechnology. Does anybody know how long it takes to double the literature in biotechnology right now? About 40 days. We double the amount of literature on biotechnologies and biology in about 40 days. Okay, if you include medical as well. Wow, right? Imagine that, a little over a month. Doubled. Yeah, try keeping up with that, huh? Okay, so we're going to have these key techniques. We have the restriction enzyme analysis, blotting techniques, DNA sequencing, solid phase synthesis of nucleic acid, polymerase chain reaction. This is typically shortened as PCR, and bioinformatics. You may have heard of PCR a lot and DNA sequencing a lot lately. They've been ones that have come up a lot because of COVID. Okay? If you're wondering why the PCR techniques, why COVID testing was such a pain in the ass and we had so many problems, that was PCR. Okay? There was a lot of problems with that during COVID. The restriction enzymes are going to be split DNA into specific fragments, and this is why they're useful for these things and why they've contributed to this great amount of progress. The restriction enzymes, again, that restriction endonucleases, in other terminology, are the prokaryotic enzymes that recognize specific base sequences and double helical DNA and cleave both strands at specific places. You may have remembered that we actually talked about restriction enzymes in the previous chapter 4 and such, right? Okay. And so the recognized sequence is almost always palindromic. That means the same forward and backwards. We actually saw that before. You remember where we had the repeated, where it had the same sequence of 3 on both sides, like a mirror image, right? The sequence is going to be recognized by Streptomyces anchromogenes. This is the cleavage site right here, and you can see the cleavage site is going to be mirrored on the other side as well. You'll have a symmetry axis, and you'll have that twofold symmetry. These words may sound familiar because we had talked about them before with a nicer looking example. Okay? Does anybody have any questions about that? Okay. See your mirrored site, palindromic site. Okay. All right, so the specificities of some of these restriction endonucleases, your names are going to consist of a 3-letter abbreviation for your host organism. That's that 3-letter abbreviation right there, see them? So that's why E. coli always has ECO in front of it when you see these different restriction enzymes. A strain designation if you need to. You don't always need to designate strains because sometimes there's not multiple strains, right? A Roman numeral to distinguish multiple enzymes from the same strain. You can have multiple different isozymes and enzymes from the same strain, right? We'd actually mentioned some of them before, but you may not have caught them because they're kind of they weren't really that important at the time. Okay. Anyways, these are some different examples and the codes that they actually would target, their specificity. Anybody have any questions about that so far? Fantastic. Fairly clear once you see the rules, right? But without the rules, it doesn't make any damn sense at all. So what are we looking here? What does ECO stand for? Yeah, right? The host. What does this stand for? Yep, you got it. And then this one stands for? 1. Yeah, exactly. Yeah, the enzyme 1, right? Okay, fantastic. Okay, so the restriction fragments, these can be separated by gel electrophoresis and then visualized. And we actually talked about gel electrophoresis in chapter 4, right? So now we're coming back, okay? And we're looking at from a different perspective. So the DNA fragments will be generated by restriction enzyme digestion. These can then be separated by gel electrophoresis. We can do that in agarose or polyacrylamide gels. They can be visualized then by staining the DNA binding dyes. Ethodium bromide, is going to be one of the more common ones that's extremely effective. Okay. I don't think we do this here though because it's quite toxic as well. Yeah. Did we do any DNA staining with bromide? Yeah, protein, right? It can be, but there's several blue dyes. Because blue shows well. That's okay. It's okay. Yeah, the bromide is pretty toxic and takes special care when you're disposing it. So they don't like to use those law colleges. It's really common not to use them now. It used to be common to use them because you had no other choices. Yeah. Probably not, yeah. Yeah. They also used to make you make your own gels, the polyacrylamide gels, back in the day, but again, takes a lot of time, very dangerous. Yeah. So those things have gotten phased out. And you can buy gels now, it's not So southern blotting and northern blotting. Yes, southern and northern. Damn Yankees. So southern blotting is going to be a technique by which a restriction fragment containing a specific sequence can be identified by hybridizing it with labeled complementary DNA strand. Northern blotting is going to be the analysis technique for an analysis of RNA. So guess what? Southern won on this one. Okay, we got the DNA. The Northerners got the damn RNA. They depend on us, right? I know. So your DNA probe, this is going to be a short stretch of single-stranded DNA, which is going to contain a known base sequence. This will hybridize with a restriction fragment having a complementary sequence. And so literally it's a probe that will bind well to what we're looking for, right? Which is nice, because that's what probes usually mean, doesn't it? So these steps in southern blotting are going to be step 1, separate a mixture of restriction fragments by electrophoresis through typically an agarose gel, though theoretically you could use a number of different gels. Step 2 is going to be the transfer to a membrane composed of nitrocellulose or nylon. In case you're wondering, yes, we do actually have nylon membranes in these labs, okay? But we haven't used them in a long time, which is sad. Yeah. Step 3 is expose the membrane to a 32P labeled or fluorescently tagged DNA probe. Remember, the labeling or fluorescence tagging is so you can later see them somehow, okay? Step 4 is going to be perform an autoradiography or a fluorescence imaging to reveal the position of your restriction fragment probe duplex. So you have a labeled probe. The probe binds to what you're looking for, okay? And the autoradiography is going to be for detecting the 32P. The fluorescence imaging would be the fluorescently tagged ones, okay? It's not interchangeable there. This is, again, southern blotting. What is southern blotting again? We're looking for DNA fragments. Very good. So here we have our DNA fragments. Why are they fragments? Exactly, we digested them. Do we necessarily have to use one restriction enzyme? No, we can use a mixture. So we then run electrophoresis. We're going to transfer the DNA by blotting. This is why it's referred to as blotting, by the way. So we blot it onto a sheet. In this case, we use nitrocellulose. This is then going to have a 32P labeled or the fluorescent probe, whichever one works in your lab. Obviously, the machine will dictate that usually, and cost. Okay, and then we're going to use the correct method to be able to visualize that probe. And we can see right here, this is the DNA fragment that was revealed, and that's the DNA fragment we were probing for, right? Does anybody have any questions? Okay, fantastic. So DNA can be sequenced by controlled termination of replication. So the controlled termination of your replication, another name for this is Sanger dideoxy method. Sanger dideoxy method is actually, in some ways, the more revealing name of this. But anyways, I've normally heard of it as controlled termination of replication rather than anything else. Okay? That tends to be the more common in my experience. The technique for DNA sequence, the fragments are going to be separated by capillary electrophoresis. Remember our separation techniques. The fragments are going to be detected via their fluorescence. The sequence of colors is going to give us our base sequences. So right here we have a primer and a template. And so what we'll do is we need to add the DNA polymerase because we need to replicate, right? We're going to use the normal mechanism for replication. So again, DNA polymerase 1. We're going to then add also excess unlabeled nucleotides. Why do we need excess, by the way? If we added just... Yeah, if we only add just the right amount, this would go incredibly slow, right? Yeah. So excess. We're then going to add fluorescently labeled dideoxy analogs. This is why that's more revealing. They're dideoxy. 2 oxygens removed instead of 1 oxygen removed, right? So instead of deoxy ribonucleic acid, dideoxy. Okay? This also is one of the reasons why this excess is important. If we didn't put excess in the first one, the normal stuff, then we would get these incorporated all the time, right? But if we put these ones that can't continue the reaction as a minority, then most what happens is we'll get a little bit of it stopped here. We'll get a little bit of it stopped the next one. We'll get a little stopped the next one, right? And eventually we'll kind of partially run out, but most of our species is going to complete it, right? Okay. And so we get a fairly consistent yield of each of these. There'll be a little fluctuation, but not huge, right? If we put basically a 1-to-1 ratio here, we would get ever decreasing populations, right? And at the end of this, we couldn't detect the last ones. Okay. Okay, so this is what we'll actually get. We'll then get strand separation. This is literally melting the DNA in one of the methods we talked about where we separate the strands. And then, so we'll get the complete one, right? And this is the complete one right here. And then what we'll get is species where they end at the dideoxy form at each ending. Everybody see that okay? And because these are fluorescently labeled, they're each going to have their own color under fluorescence, and they'll show up like so. And again, this is the elution time from the capillary. That depends on the size, right? Okay. Anybody have any questions there? No? Okay. Fantastic. Okay, so the next one's going to be controlled termination of your replication reaction components. The controlled termination of replication reactions will require these ingredients, as it were. DNA polymerase, a primer, a template, the template being the DNA template strand, i.e. the one that we want to be sequenced. The 4-deoxyribonucleoside triphosphates, because we'll need ingredients to make this from, and a small amount of 2', 3' dideoxy analog of each nucleotide, each with different fluorescent labels. Again, the dideoxy is not going to allow the additional nucleotides to be added, right? Yeah. Okay, and so this is actually the dideoxy form. Can everybody see that okay? The 2' deoxy like normal, right? But the 3' also deoxy. So we got 2 and 3 deoxy, don't we? 5' still can work. See that? And so we can continue attaching the 5' to the previous 3'. But when we try to attach the next 5', we have no option. Can't do the reaction. Okay. Again, because we're putting in a minority of these, we're going to generate fragments of different lengths. The majority will get to the end and make the full fragment, right? Okay, and then this is going to incorporate, I think we're good. Anybody have any questions about that? No? Okay, yeah, it's kind of important to understand the techniques surrounding it. I think you guys got it. Okay, so our DNA probes and genes, these can be synthesized by automated solid phase methods. The oligonucleotide synthesis can occur by sequential addition of activated monomers to a growing chain linked to a resin. The activated monomers are going to be protected deoxyribonucleoside 3' phosphonamidates. The newly synthesized DNA can then be tagged with 32P or a fluorescence marker. Some of the possible applications of this methodology is going to be using it as a probe, kind of like what we had just discussed. We can also use this as a primer. We can synthesize new tailor-made genes using this method as well. Keep in mind, because they are linked to a resin, we have them mostly immobile, and so we can wash things over them, right? And control their conditions much more closely. So this is again a deoxyribonucleoside 3' phosphoramidate. More water. Excuse me. This is with DMT and beta CE attached to it. This is the beta CE, by the way. Full name, beta cyanoethyl, and then it's a group, right? Notice the name actually says what it is. This is attached to the beta carbon, right there. This is the cyano group. Okay? That's the organic chemistry, isn't it? This right here is DMT. That's the same DMT we've talked about in organic chemistry as well. So that's that dimethoxy trityl. Again, this is going to be attached to the 5' OH group right there. This is going to act as a protecting group. Right here we have our nucleoside right here. This right here is then the 3' oxygen. And again, we have that rendered on-reactive via this attachment of this group right here, which is the beta CE group. Anybody have any questions? Okay, so the oligonucleotides, these are going to be synthesized in your solid phase by the phosphite triester method. So right here we have the oligonucleotide. Again, this is that solid phase because we have attached it to a resin, right? And so it can't move around, but we can remove the different reactants we want to expose it to, different conditions we want to expose it to through our columns, right? Okay. Technically, you don't need to use column, but yeah, column anyways. So right here we have the base, which is N-1. Here we have it, the growing chain right here, which we're going to attach this to, okay? And so we're going to then attach this via coupling right there. Again, this is the phosphate triester intermediate right here. And you can see again, that's that that phosphate triester right there. See that? Okay. Well, it's not a triester yet. It has an amine on it. Right here, it's a triester. Okay. And see, we're exchanging that amine for the oxygen here. That was on our growing chain. Okay. Okay. Notice that is the same side as the BCE. That's why this BCE needs to be there, by the way. Otherwise, it could react on that side. Okay, so the next step then is right here. We're going to go down. We're going to perform oxidation via I2. This will give our phosphotriester intermediate, which is this form right here. You can see here we have attached that carbonyl group, right? That's the oxidation. Okay. The next thing that has to happen is we still have that protecting group, DMT, that is causing this 5' end to be on-reactive, right? So we need to remove that protecting group so we can expand the chain further, okay? And so what do? Well, we deprotect it. We're going to use dichloroacetic acid. That'll remove the DMT, just like we had talked about when we talked in organic chemistry about protecting groups, right? And then what's going to happen is now we have a new 5' end exposed. We now add whatever amino base we want to add to the next one. Okay? And so we do the process again. Repeat, repeat, repeat. We'll specifically use species with whatever base we want to add. Okay? And that's why we can target it. This is exactly what we want it to be. All right. Okay. Does this make sense? Did anybody have any issues, questions? No? Makes sense? Yeah. Basically, it's a method that we have a fixed chain, and we can flow across whatever amino acid we want and selectively react it on. Okay? Not amino acid, God. Base. The protein one that we did. Freudian slip again. Proteins. So the selected DNA sequence, these can be greatly amplified by the polymerase chain reaction. This is polymerase chain reaction, typically called PCR. This is the method for amplifying specific DNA sequences. The PCR reactions are going to require a pair of primers that will hybridize with flanking sequences of the target. All 4 deoxyribonucleoside triphosphates, i.e. your DNTPs, ATP, CTP, everybody got that? And a heat-stable DNA polymerase. You need this to be running at a temperature it would not normally run, and so you need this to be heat-stabilized. An interesting side note to this is the graduate student that worked on this took about 8 years to graduate because he couldn't publish anything for a long time until he figured out how to do this. And so it took a really long time, one of the longest times ever recorded for a successful graduation in PhD. He retired when he graduated because this was a huge commercial success, right? There's PCR machines in every hospital everywhere, okay? There's PCR machines in all vaccine production and everything, okay? And the PCR is actually one of the reasons why we couldn't do the testing for COVID right away, is because one of the tests was PCR, and we didn't have enough PCR machines and people to operate them. So it slowed that whole thing down. So a PCR cycle, the PCR is going to happen in cycles. Your PCR is going to consist of 3 steps. We'll repeat that 20 to 30 times typically to get our amplification. So step 1 will have 2 strands of parent DNA. These are going to be separated by heating the solution to 95 degrees Celsius for 15 seconds. This temperature is actually very important. Going too much high or too low will greatly affect your PCR rates. Okay, this actually was a big problem because if you don't let your machines cool down and have rest times, you can have trouble controlling temperatures. Anyways, so what's happening here? What do you think? If you heat like a solid up, what do you get? Nothing. So you're melting the DNA. You're causing the hydrogen bonds to separate, causing the 2 strands to come apart, right? Okay. So step 2, the solution is then going to be cooled to 54 degrees Celsius. This will allow the primers to anneal to the DNA. And so what we'll have is little fragments that prime it, right? The primers will stick on. Step 3 is then going to have the solution heated to 72 degrees Celsius. This will allow the polymerase to elongate both the primers in the direction of the target sequence. So we stick the primers on, we heat it back up, and allow the polymerase to start running. Okay? By the way, 72 degrees Celsius, that's not physiological body temperature, is it? No, that's quite a bit higher, isn't it? This is why it has to be this heat-stabilized polymerase. It's also why it happens so much faster than your body does. We're doing it at a higher temperature, right? After n cycles, the desired sequence will be amplified by a multiplication of 2 every cycle, right? And so if we do 20 cycles, that would be 2 to the 20th power, which is a lot of amplification, isn't it? This is why you can take a few DNA strands, few DNA fragments for something. You can amplify it enough that you could do almost any testing you can imagine, right? Yeah. And that's why they use this for like detecting DNA at crime scenes or many, many, many purposes, right? Figuring out what sort of DNA you had from a little drop that you sent to them on a cardboard thing. Cloning you, maybe. Hopefully not cloning you. That's bad. This is the first cycle of PCR. Again, we'll have the flanking sequence, the target sequence. We're going to add excess primers, heat to separate them. And so we separate the DNA strands with the primers in there. We're then going to cool. Those primers are going to anneal on the flanking sequences. The flanking sequence being the ones that are just off of what you want to target. Okay? And so then they attach to the parent DNA. That puts primers on them. Now, once we heat this back up, the polymerase is then going to start activating. It will attach to the primers and synthesize new strands of DNA from those primers on. Okay? Notice we do not just get the target sequence. We get the target sequence and then down, right? Okay. This is then going to be done in multiple cycles. Notice that our previously made DNA is also going to replicate as well, right? So we have an exponential relationship, exponential effect, okay? After 20 to 30 cycles of this, you get a huge amount. Yeah. And you could run this basically as much as you want. Now, let me ask you a question. Is it possible to get the exact DNA fragment out perfectly every time? No. There's going to be error, right? Same as there is in you, there'll be mutations, okay? And so they've actually had cases where they like mutated species and cloning and stuff because of this. Yeah. So you do have to watch out for this, okay? The PCR, it's a powerful technique in medical diagnosis, forensics, studies of molecular evolution. This is a hugely wide used technique. There's just a massive variety of applications for this because we replicate DNA fragments, right? And so any testing that we want to do, instead of having one droplet to try to test and, oh shit, we destroyed the one droplet, we're screwed now. We have basically as much as we want, right? Will it have evolutions happen if you keep replicating the same DNA? Yes. Which is why you normally take very small fragments of DNA and you store any extra, okay? By the way, store extra. One of the most important things I can tell you, yeah, don't use all of your original sample. Yeah, use aliquot, small amount, store the extra somewhere where you can get to it. Because there's going to be a time where you fuck up. Okay? You don't store that extra. It's going to cost you. All right. So diagnosing certain infections caused by viruses and bacteria, this is probably the most famous now because everybody went through COVID, right? And so if you look back in the COVID era reporting, you'll see a lot of discussion over PCR, okay? Detecting certain cancers early and monitoring chemotherapies, because again, you can get a small draw of the blood with just a few DNA fragments from that cancer, and then you can replicate it up until you can detect it, okay? Solving forensics cases, this is probably the most famous before COVID, okay? You know the DNA testing, like Project Freedom and stuff? Yep, that's it right there. By the way, DNA testing is not perfect. Anybody that says it's perfect and proves cases on its own, no, it does not. Actually, DNA testing has actually had false convictions, and vice versa. Okay. Studying molecular evolution. This is most of the molecular evolution now that's being done is being done by a PCR of some sort. Okay. The tools for recombinating recombinant DNA, technology, have been used to identify disease-causing mutations. Polymorphisms, these are instances of genetic variation. These may cause disease or be closely linked to the responsible genetic alteration. Restriction fragment length polymorphisms, which are abbreviated RFLP. These are polymorphisms within the restriction sites that will change the sizes of your DNA fragments produced by the appropriate restriction enzyme. So powerful means of correlating a mutation with the occurrence of a disease, because it gives you a really, really easy to detect marker, right? So our previous testing, if you have a different size, it's actually incredibly easy to detect, isn't it? Okay. Whereas if it does not change the size, it is harder to detect. We'll have to do more work, right? So again, this technology has actually revolutionized basically all aspects of biology. This is one of the major reasons why we've seen so much development over like the last 20 years or so. Okay. The vector, anybody know when PCR was invented, by the way? It started showing up loosely around like 1980s. Yeah, it's not a super new technology. It's gotten better and more automated. But recombinant DNA is much newer. This is something that started only really showing up in the 2000s. Okay. A vector is going to be a DNA molecule that can replicate autonomously in an appropriate host organism. It enables the rapid covalent insertion of DNA fragments that we're interested in. Or if it's HIV, it just does it on its own. Shit. The choice vectors for cloning in E. coli, for example, include plasmids. These are naturally occurring circles of DNA that act as accessory chromosomes in the bacteria. The virus bacteriophage lambda, which is K-phage, is what the abbreviation typically is. E. coli is very, very common to incorporate these recombinant DNA strands into, because it grows very easily and it's well understood how to maintain it. Okay. So inserting DNA fragments into vectors. Step 1, you'll cut the vector open. So you cut the vector by the restriction enzyme to create cohesive or sticky ends, because we need to insert something in it. First step, cut it open, right? What do you think sticky end might mean? Exactly. Yeah. It typically means that it's not a straight cut. You have one side is a little bit longer than the other. Yeah. Exactly. So you can get a match, right? Or like Tetris. Yeah. Step 2, insert the DNA fragment within the cut vector using DNA ligase. Step 3, add linkers if needed to yield cohesive ends so that you can rejoin it. This is the overall graphic for it. So again, different DNA molecules can be joined by the cohesive end method. So right here we have a PSC101 plasmid. Remember, this is the accessory DNA in E. coli usually. This is then cleaved with EcoR1 restriction enzyme. And we end up with sticky ends here. See that? They're sticky because you can make the hydrogen bonding and the hydrophobic interactions, right? So you have a little bit of energy for inserting it, okay? Without the sticky ends, it's like trying to glue really smooth surfaces together. It's really hard, right? Okay, this right here is then going to be our DNA fragment we're taking from our target. So we're going to, again, cleave that with EcoR1 restriction enzyme. Does that mean that we may not get exactly just the target we want? Yes. Yes. Then why do we use EcoR1? What has to match? The sticky ends, right? If our sticky ends don't match well, what's going to happen? Yeah, we're going to have a lot of trouble sticking, right? Yeah. And so that's why you typically cleave this with the same restrictive enzyme. Okay? So that your ends will stick. Because they match, remember? They match those sequences of 3 on each side. Okay, so the annealed, you're going to then anneal the DNA fragments. They'll rejoin with DNA ligase. If you just anneal them, they will actually stick, but they will not permanently insert because it will just be hydrophobic interactions and hydrogen bonding, right? And so they can come apart very easily. The DNA ligase is needed to do the covalent modification on the backbones. Okay? And that's what DNA ligase does in the normal circumstances as well. So, the linkers can be added to yield cohesive ends. Linkers are going to be short, chemically synthesized DNA segments that will contain specific restriction sites. You may have went like, well, does that mean we can only do things where the target would be what the restrictive enzyme can target? No. We can do some more work, okay? i.e. make our own ends. So again, these linkers are going to be those short chemically synthesized DNA segments. They're going to contain the specific restriction site. They'll be added when the target DNA fragment is not naturally flanked by your appropriate restriction sites. Okay? They're going to be covalently joined to the ends of the target DNA fragment using T4 ligase. Remember that previous method we just talked about where we could synthesize short fragments of DNA that we want? Guess what? That's one of the ways you can get these, right? You can specifically synthesize little 6 member fragments and then use them as linkers, right? Okay. Again, that's the T4 ligase. The cohesive ends are going to form when the linkers are cut by the restriction enzyme. You oftentimes actually will make these where it'll have like a set of restriction enzyme places where you can cut them for different restriction enzymes. And they can actually sell these, which is kind of interesting. Okay? So you'll have like an ECO R1 restriction and then another restriction enzyme and another restriction enzyme. That's the whole DNA fragment. It's just different restriction enzyme spots. Yeah. Okay. So the formation of cohesive ends is right here. Right here we have the DNA fragment or vector we're looking at. We're going to join our blunt ends with the T4 ligase. This is why we have to use that specific T4 ligase. So we join the blunted ends with our linker. See the linker right there? Okay. What does decameric mean? Meric polymer, deca meaning? Yeah. Got it. Okay, and then so we link these on both sides. Then we're going to take whatever restrictive enzyme we used to cut the previous one, right? The plasmid. We'll cut this with it. And now we have matching ends. So we have matching sticky ends. Then we can put that in there, and congratulations, we can join it like we talked about here. Anybody have any questions? No? Fantastic. Okay. So again, the plasmids and phage are going to be the choice vectors for the DNA cloning in bacteria. The plasmids will range in size from 2 to several hundred kilobases. Anybody remember what kilobase would be? 1,000 bases, right? Okay. So these are like several hundred kilobases. I mean, hundreds of thousands of bases. They're very long. These are going to carry genes for antibiotic inactivation, toxin production, and breakdown of natural products. I know I just said that these are very long, but keep in mind that these plasmids are actually the short DNA fragments that are round, right? They're not usually the primary DNA fragments that carry the replication of the cell. So these are going to be accessory chromosomes. These are going to replicate independently and may be dispensable, actually. You can actually fully lose them from E. coli, and they will continue functioning. Okay? Maybe not as well in specific environments, but they can continue functioning. The cloning vectors, these are going to, by the way, why do we target the plasmids instead of the main chromosomes? Why do we target the accessories? what if we cut in the middle of something important? What's going to happen? Yeah, so if it's the main chromosome, it's going to damage it, right? A lot. And it may even just kill it. If we cut into the accessory chromosomes, that doesn't necessarily need to survive. Worst that happens is it doesn't grow quite as well, right? Yeah. And if we do this a lot of different places on those, we'll get something that works. Okay? The cloning vectors, these are going to be the class of plasmids that are particularly suitable for the insertion and replication of DNA fragments. They're going to contain a polylinker region that includes many unique restriction sites and reporter genes that encode easily detectable markers. In case you're wondering, yes, there are E. coli that have specifically been evolved and grown to do this with. Okay? They're usually patented, by the way. So like a specific lab would have a patent on them. And I used to know Aaron Fenton, worked with him for a bit, and they had several patented strains that they actually sold for $10,000 a piece. So they'd grow them and sell them for $10,000 to grow your own for a little bit. Yeah. So it's kind of something. This is the cloning vector PUC18. Right here we can see a polylinker. See that polylinker right there? Okay, that little part of DNA right there, that polylinker, is literally these different restrictive enzymes targets. And so you can cut into that and put whatever you want in there using all of those different restrictive enzymes. Okay? Cool, huh? Yeah. Okay. This also has some kind of interesting deals, like this is the origin of replication, allows you to replicate this and put it into the new E. coli's. Was this E. coli? This might not be E. coli. And then, like, this is ampicillin resistance, so it has a resistance strain in here that actually will let you kill off all the other bacterias that did not have this in it. Okay? And so there's kind of cool stuff in this that you may not have realized at first glance. The component of the PUC18 cloning vector, so the cloning vector of the PUC18 will contain the origin of replication. Again, this is because you need to have that plasmid replicate when the cell replicates, right? Will this do you any good if the cell doesn't replicate making the protein or whatever you're trying to get? No. It just died off in one generation, it would suck, right? So it wouldn't really do you any good. This will also have a gene encoding the ampicillin resistance for selection. Again, you can then use the ampicillin to kill off all the stuff that does not replicate with this, right? Allowing the one that actually has this gene to thrive, okay? Without that, you would actually start seeing other strains coming in, competing, and they may actually kill off your target. Like bacterial mold, right? Or fungi. The fungi wouldn't be killed by that. The LAC-Z gene, which will encode the part of the beta-lactosidase. This right here is the insertional inactivation will disrupt genes. So insertional inactivation will be the disruption of a gene due to the insertion of the DNA fragment into the polylinker region. The beta-lactosidase cleaves the synthetic substrate X-gal. This actually releases a blue dye. Your bacterial cells containing a DNA insert within the polylinker will no longer produce the dye in the presence of X-gal. So the bacteria harboring the vectors with an insert are going to be white. The ones that are without the insert will be blue. And so what does that do for you? So normally you would not do this with one tray of bacteria. Okay? So you mix up your agarose, you put your bacteria in it, you then put a bunch of trays. Okay? And then you would try to insert the gene in all those trays. And then what would you do? Well, you'd give them ampicillin, right? Kill off everything else. You'd usually put that in with the agarose. Okay? And then a week later, or however long it takes it to grow and replicate, so you can see it easily, you would again look at the trays. If the trays are blue, what do you do with them? they don't have the insert, do they? So whatever you're trying to insert here, they don't have it, right? Won't do you any good growing those, okay? Yeah, you probably kill them so that they don't do anything. Because you could insert something else by accident into a different place and mutate them, so kill them. The bacteria harboring the vectors, those are going to be white, right? And so you look and you get white ones. If the majority is white, cool, I can use that. And then you can monitor that and see if the blue accidentally take it over, okay? And it's feedback, right? Like hopefully you've gotten from me by now, real science needs feedback loops, right? Okay, feedback loop. So insertional, how's my time, by the way? Oh, okay. So this is the insertional inactivation that I was just talking about. See how it cleaves the lax Z gene? That's why I was on both sides. And cleaving that's going to be causing this to turn white. If it's not cleaved and it still is active, it's going to end up having a this blue color here. Okay. Note that that does not actually mean just because you inactivated the gene that you actually inserted what you wanted to. You could insert different things. Yeah. All right. We'll stop there. You all have